Welcome to a small journey into a recent scientific study that tackles a surprisingly persistent problem in chemical regulation. How do we reliably identify substances that can cause respiratory sensitization, conditions like occupational asthma, without relying on animal testing? Despite decades of research, there is still no fully accepted and validated test method specifically designed for this endpoint, although significant developments are ongoing nowadays. And that's where this new work comes in. Let's start with the regulatory landscape. Under frameworks such as the Globally Harmonized System, GHS, and the European CLP, respiratory sensitizers must be identified and classified because they can cause severe and sometimes irreversible health effects. However, unlike skin sensitization, there is no OECD-accepted test guideline specifically for respiratory sensitization. In practice, this has led regulators and industry to rely on non-standardized human data or surrogate methods, particularly those originally developed for skin sensitization. One prominent example is the Direct Peptide Reactivity Assay, or DEPRA, a standard assay validated by OECD. The DEPRA measures how reactive a chemical is toward protein-like peptides, specifically targeting cysteine and lysine residues. And the reasoning seems sound. Sensitization typically begins with a chemical binding to proteins. This is true for skin sensitization as well as for respiratory sensitization. In fact, this process step is formalized in an adverse outcome pathway, or AOP, for respiratory sensitization. This AOP describes a mechanistic sequence of events starting with the covalent binding of electrophilic chemicals to proteins in the respiratory tract, referred to as key event 1. This initial protein binding can alter protein structure and trigger downstream biological responses, including activation of immune cells, production of cytokines, and ultimately a T helper 2-driven immune reaction that leads to clinical outcomes such as asthma. Importantly, the chemoassay presented in this study specifically targets this first step, key event 1, by quantifying the reactivity of chemicals toward amino groups as a surrogate for protein binding. But here's the problem. When it comes to respiratory sensitizers, the DEPRA does not perform reliably. So what goes wrong? The UFZ work, as presented in the publication of Simony 2025, shows that certain substance classes are not always correctly identified by the DEPRA, even though they are well-known respiratory sensitizers. The root cause is surprisingly technical, but critically important. In the DEPRA at the assay's pH, the used buffer itself substantially reacts with the test chemicals so that the chemicals cannot react with the protein-like peptide as they should. The result? Their true amino reactivity is underestimated. And since amino reactivity is a key signal for identifying sensitization potential, the standard DEPRA effectively masks some of the most relevant substances. Now let's turn to the proposed solution. The researchers of UFZ introduce a new chemoassay based on glycine paranitroanilide as a model for amino residues in proteins. Conceptually, this is very similar to the DEPRA. It can measure how much of the model compound is depleted over time. So from a regulatory and scientific perspective, it's not a completely new paradigm, but rather a refinement of an existing one. The key difference lies in the experimental setup. The new chemoassay uses a different buffer at physiological pH. This seemingly small change has a major impact. It avoids unwanted side reactions and it provides a more accurate measurement of intrinsic amino reactivity. From a practical standpoint, the method is also straightforward, standard laboratory setup and simple reagents. In other words, it retains the simplicity and accessibility that made the DEPRA attractive in the first place. Now to the key question. Does the new chemoassay actually work? The answer appears to be yes. The study tested a range of chemicals comprising a broad range of reaction types, both known or suspected respiratory sensitizers and non-sensitizers, taken from earlier reviews. Using the new chemoassay on the left, nearly all potential respiratory sensitizers showed high amino reactivity. Non-sensitizers consistently showed low reactivity. Importantly, this resulted in a clear separation between the two groups. This is in stark contrast to the DEPRA on the right, where both groups often overlap. In practical terms, this means the method correctly identifies a large proportion of true sensitizers. The only known limitation of the new chemoassay is that it cannot be used for proreactive compounds due to missing transformation capabilities. This is exactly what is needed for a reliable method in regulatory contexts. So what does this mean for GHS and CLP frameworks? The authors are careful not to overstate their conclusions. The new chemoassay is not yet a validated test guideline. However, with its promising recognition power, as indicated in the marked cells of this table, it has strong potential as part of an integrated testing strategy, an IATA. In such a framework, it could: first, identify known respiratory sensitizers with mechanistically relevant evidence based on their protein-binding reactivity. Second, fill a methodological gap for respiratory sensitization, especially for the first key event in the respective AOP. Third, complement in vitro methods addressing further steps of the AOP of respiratory sensitization. And perhaps most importantly, it offers a non-animal approach that aligns with current regulatory priorities. To sum up, this study doesn't reinvent the wheel, but it significantly improves how the wheel turns. By refining the DEPRA concept, the new chemoassay may be used as a practical, mechanistically sound, and regulatorily relevant method for identifying respiratory sensitizers. Thanks for joining this small journey.